Bismillahirrahmanirrahim. Welcome again, guys. Welcome to another video lecture by Kami Biology. Today in this video lecture we are going to talk about elongation of DNA replication in detail. Okay, so let me write elongation of DNA replication in eukaryotes. In the previous video lecture, if you guys remember, we talked about proteins that are involved in DNA replication in eukaryotes, and we talked about initiation of DNA replication in eukaryotes. Now, in the previous video lecture, we are at that stage, means the two strands are completely separated from each other and it make a replication bubble, as you can see. So this replication bubble is formed when the two activated complex, which we call as CMG. Inside the CMG we have CDC 45 MCM and genes, so they make activated complex. Due to this way they can melt the DNA by breaking the hydrogen bond between the DNA strand, and due to this way we get a replication fork. So two replication forks is moving in opposite direction, due to which we can get the replication bubble. Two strands completely are separated from each other in the form of replication bubbles. Now, once we get the replication bubbles, then we talk that DNA primase or primase enzyme is activated and they can add the primer, RNA primer. A few stretch of the RNA primer they can add here. They can add just one RNA primer at the upper strand, which we call that leading strand, and they can add multiple of the primer at the lower strand, which we call that leading strand. Now, moreover, this then we have another enzyme which we call that DNA polymerase alpha. DNA polymerase alpha can add a few stretch of DNA nucleotide, not RNA, DNA nucleotide, and it may be two to three DNA nucleotides they can add. As you can see, this blue one represent the DNA nucleotides. Now it can be added by the DNA polymerase alpha. Why? Because DNA polymerase delta and epsilon can start the elongation process. But they can start the elongation process when there is DNA nucleotide present. DNA nucleotide will be present with the RNA primer, and this DNA nucleotide will be provided by the DNA polymerase alpha. Now, once all this process is done, so then DNA polymerase delta, as you can see, this red one is activated. But before that, this PCR ring is activated and they can bind on both of the strand, as you can see. Now, once they are bound on the both of the strand, then what are the function of this PCR ring? This PCR ring is very important, because this PCR ring can hold the two strand, and they can also act as a sliding clamp for the DNA polymerase, and they can also hold the DNA polymerase. The weight of the DNA polymerase is very high, that's why there is a chances that DNA polymerase can fall, so it can prevent the DNA polymerase from the falling. So that's why PCR ring just hold the DNA polymerase. So this PCR ring is very important, and they also push the DNA in this direction and in this direction as well. But remember, DNA polymerase can add the nucleotide in 5' to 3' direction. Okay, whenever the elongation process is started, so in this case DNA polymerase must edit the nucleotide in 5' to 3' direction. This is very important. So at the upper strand we have the direction is 5' to 3' direction, so the upper strand will be synthesized in 5' to 3' direction. So there is no issue. So only one RNA primer we need and a few of DNA nucleotides, so then the DNA elongation process will be continuous, which can create no problem. But remember, at the lower strand there is a problem. Why? Because at the lower end the strand will be synthesized in 3' to 5' direction, because the lower end having 5' to 3' end, so the new strand will be synthesized in 3' to 5' direction, which is the complementary. So it will be synthesized in a segment form. Why? Because DNA polymerase need a free hydroxyl group. As you can see, this blue one having the 3' hydroxyl end. So that's why. So it will be present in a form of segment form. So DNA polymerase delta can hold the primer, or it can hold the DNA nucleotide having the 3' hydroxyl end and then start the elongation. just like that. So that's why it can, this DNA polymerase can start the elongation process at the lower end. They can start the elongation process in a segment form. Okay, so this lower strand will be synthesized in a segment form, which we call it Okazaki fragment, and this strand is synthesized in a discontinuous manner. That's why we call that discontinuous strand or lagging strand. Okay, now let me explain that what actually happened. If I draw the same structure here, so again we have, so let's suppose this is a replication fork, and there is also a replication bubble, and another replication fork is also present, but I'm not going to draw it again here. So let's just draw here one replication fork and one of the CMG complex to explain the whole process of the elongation. So again, this is the MCM ring. They have GINS protein, as well as they have CDC45. Now, and it make the CMG complex, and then it can move in this direction. Now remember, at that specific point, the RNA primer is added. Okay, and some of the DNA sequences is also added. Some of the DNA sequences, which is added by the DNA polymerase alpha. And after that, pCNA ring can bind here, and they can start the elongation process, as you can see. Okay, so this is delta. Now the elongation process is start, and this strand will be synthesized in this direction, which is 5' to 3' direction. Okay, but at the lower end we have multiple of the segments. And remember, this lower strand make a loop. This lower strand make a loop. Now, why it can make this bending? Because they need the same direction. Okay, so the direction will be the same, 5' to 3', but actually the lower strand is synthesized in 3 to 5' direction. If you guys remember, we also talked in prokaryotes, there is also loop formation in the lagging strand. So in this eukaryotes, the loop is also formed, means the DNA is bent just like that. Now why the DNA is bent? Because we need a same direction. So when a loop is created, so remember, due to this loop formation, the direction is same. Okay, so if this is a 3' to 5', let's suppose, so similarly the direction is changed, as you can see. Okay, that's why loop formation is very important. Now then again at that here, now at here the pCNA ring can bind, as you can see, and the DNA polymerase delta can also bind with this pCNA ring and start the process of elongation. Okay, then the process of elongation will be continued. So they elongate this strand. But remember, in this case, what actually happened? When they start adding nucleotide, as you can see, so nucleotide addition will be continuously in discontinuous form. And when it reached to this end where the phosphate group is present and hydroxyl end is also present at that specific point, then what actually happened? This DNA delta can remove a few stretch of the RNA nucleotide, as you can see. So it remove a few stretch of RNA nucleotide. 2 to 3 or 4 nucleotide are removed, and they can add the nucleotide, and a few RNA primers is also present. So we get a flap. So a flap is formed, as you can see. Okay, now this flap should be removed by another protein which we call that fin. Fin means flap endonuclease protein or enzyme. It can come and remove this primers, the remaining primers it can remove, as you can see. Okay, and then it can add the nucleotide. So the nucleotide addition are now continuous. Now remember, when they reach to the primer, let's suppose they reach to that primer, then what actually happen at that specific primer, if they reached here, let's suppose then they leave the strand or they leave this primer. When, let me once again tell you, when the DNA polymerase delta leave at that specific area, then they leave the primer. Why? Because at that specific point we have the phosphate group. Okay, so they leave the primer and the strand is now elongated. Then they hold another primer. Let's suppose they hold another primer from that site. Because DNA polymerase need the hydroxyl end, and this hydroxyl end will be present at the 3' site. So they add another primer where the hydroxyl side is present, having the DNA nucleotide is also, as you can see, and then they start the elongation process again, as you can see. And then when they reach to this one primer, then they leave it, and then they hold another primer having the 3' end and add the nucleotide, as you can see, and then leave it, and then they hold this one primer and add the nucleotide addition, DNA nucleotide addition, and then leave it when they reach to this one primer. And this process is continuous. And if you can see, so the DNA synthesized in 3' to 5' direction. Okay, so the upper strand there is no issue because there is the direction is same, so they can add the nucleotide in a continuous form. That's why this strand is called as leading strand, leading or continuous strand, while the lower strand is called as lagging or discontinuous strand. Okay, now remember, this process is continuous. Okay, but these primers should be removed, and these primer are actually RNA primer, so it should be removed because in DNA we have only DNA nucleotide. So this primer should be removed, and this primer is removed by another enzyme we call that ribonuclease, or we call that RNase H. So it can remove the RNA primer. So remember, when the RNA primer is removed, as you can see, so then a gap is formed, as you can see. Okay, these gaps are formed. Again, these gaps is filled by DNA polymerase delta, so they can fill the gap by adding correct nucleotide. Okay. Now, here only one primer is added, as you can see. So one primer is removed, and the gap is filled by the DNA polymerase delta, or usually DNA polymerase epsilon at that upper strand. Okay, and then add the DNA addition of the nucleotide. But remember, they will just fill up the gap. Still the nick is present, as you can see. The DNA strand is found, but here the nick is still present. What does nick and gap means? Nick means that the two strands are bind with each other, but the phosphate group is not present, or the phosphodiester bond is not present between them, and phosphodiester bond should be needed to complete the strand. And DNA polymerase cannot they cannot add the phosphodiester bond, they just add the nucleotide. So this phosphodiester bond will be formed by another enzyme we call that DNA ligase. So DNA ligase can just add what they can just seal the nick between the two strands, and once the nick are sealed, so then the process will be completed. So remember, then this fork is moving again in this direction and open the strand. But remember, whenever they are reaching at that direction, then there is also a supercoiling is also happening, as you can see. So there is a supercoiling, and this supercoiling should be removed. Remember, when the fork or when this complex is moving, so the supercoiling is created, and this supercoiling should be removed, or the tension will be relieved. This is called tension or supercoiling. Now this tension will be removed by another enzyme we call it topoisomerase. So at that specific point, the topoisomerase can bind, as you can see. Okay, now topoisomerase can release the tension. They can cut one of the strand and join it with the same strand. They can make different cut in the strand, as you can see. So when different cuts is created in the supercoiling of the strand, so then the tension is automatically released. So when the tension is released, so again the strand is, so this is the strand, so the tension is released out, as you can see. So the complex is again moving in this direction, and the strand is opening once again. Okay, this is how the process is going on. Now remember, when this fork is going in this direction, so this two strand will be although these two strand are synthesized, let me draw here once again. So this is the replication fork, and let's suppose these are the newly synthesized strand, as you can see. So again it is coiled around, as you can see. Okay, and this is the complex which is moving on. Okay, so whenever the complex is moving on and whenever the new strand are synthesized, so it again coil with each other, as you can see, and this complex is moving until it reach at a specific point where the elongation are end and the termination process is started. Okay, and DNA replication ultimately stopped, and the two strands are formed, and it is separated from each other, but we will discuss it in the termination. So here one thing that you should note here, that whenever the new strand are formed, so they again coil around with each other. This complex need energy, and this energy is provided by the pyrophosphate, or we can say inorganic phosphate can provide the energy to this complex, due to which the complex can move and it can melt the DNA, means it can separate the two strands of the DNA. So that's why they can need the ATP energy from this pyrophosphate. So the 2-phosphate means if this is the one of the nucleotide, so other nucleotide are present here, and there is the 3-phosphate are present. So these 3-phosphate are 2-phosphate are removed out, due to which the energy is released, and due to this release of the energy, the complex is moving on. Okay, this is how the elongation process is continuously carry on, but continuously the replication, this strand will be synthesized in a continuous manner, while the lower strand will be synthesized in a discontinuous manner. And that's all about the elongation of DNA replication in case of the eukaryotes. I hope you guys get the idea about the elongation of DNA replication, but if still there is any confusion in your mind, you can write it into a comment. Inshallah, I will give you answer as soon as possible. Inshallah, in the next video lecture, we are going to talk about the termination of DNA replication, and to complete the DNA replication in eukaryotes. Okay, so keep watching the videos. Thank you so much.